The mystery of hearing. Welcome to the second part of our hearing story, second lecture about hearing. And this lecture is about what happens in the brain after the signal was detected by hair cells in the ear. Watch the previous lecture if you need to know about the ear and the cochlea. So, as a reminder, in the cochlea, things vibrate, and different parts of the cochlea vibrate with different frequencies. This is an actual cross-section through an actual cochlea. It's from a mouse, so the cochlea was cut and put under the microscope, and a sound is played to the cochlea. And you can see part of it vibrate. These are hair cells, and we can see outer cells in these triads, like these would be outer cells, and these are inner cells. As you remember from the previous lecture, outer cells act as amplifiers. They amplify the frequency sensitivity of different parts of the cochlea, and inner cells are talking to the brain. And you can see how the entire thing vibrates. And actually, if you look carefully, you can see that the outer hair cells vibrate kind of a little bit more eagerly than the entire thing as a whole, like you can see it here. They don't just vibrate passively with the cochlea, they actually resonate with the vibration and shake it back. And that's what gives it this resonance, this tight frequency tuning of different parts of the basilar membrane. But so what happens now is we have this mechanical, or rather electromechanical, separation of different frequencies that make up the total sound, and each part of the cochlea now vibrates with its unique frequency, and inner hair cells that are located here are activated when the frequency is present, and they are ready to send signals to the brain. So from the hair cells, from inner hair cells around here, there would be axons. It's not actually growing from the hair cell itself. There is a synapse here, there is a neuronal body here, and the axons go to the brain. And the neuronal bodies also run along the cochlea and form a spiral ganglion. But now we have a question. So each part of the cochlea resonates at a certain frequency. So each neuron only deals with one oscillation at a particular frequency. This is the oscillation, which is a sine wave, and that's what each neuron in each part of the spiral ganglion deals with. Different neurons would deal with different oscillations, but each neuron either has something like that when this frequency is present, or has a flat line, nothing vibrates, nothing happens if this frequency is absent. So when the frequency is present, how should this neuron spike? What information it can send into the brain? And by how it should spike, what do I mean by that? If you were Archangel Gabriel, again, if you were to reverse engineer the brain, how would you make it spike? Because look, we want the brain to have as much information as possible, right? We had to disassemble the sound from one waveform into this spectral form, into information about which frequencies are present. We had to do that because we could not use spiking to communicate the waveform of sound as your computer does it, because computer is just much faster. The computer can't just really communicate different pressure values so that the speaker would move accordingly and reproduce the sound as it was. The brain cannot do that. Our processes are much slower. Our neurons are much slower. So we had to do this disassemblage. Is it the word? We had to disassemble the sound into all the frequencies with amplitudes and communicate them separately. So what would this neuron communicate about its frequency? It's responsible for this frequency. What will it communicate? I mean, in the simplest case, it can just spike when it's present and not spike when it's not present, right? So we always have that. But can we communicate more? So think about that. If you may be not an Archangel Gabriel, all right? Maybe you're like a low-level angel who works in the Archangel Gabriel Construction Bureau. So Archangel Gabriel was asked to develop cochlea, but your goal is just to develop this tiny little neuron. You want to communicate as much information to the brain as possible, and then you hope that some other angel who's working on the brain part will figure out how to deal with this information. So we're not concerned with this part so far. Let's just make sure that we send as much information as we can, all right? And then just let's hope that the brain will figure it out somehow. that evolution or another group of angels, like actually it's evolution, but let's pretend that there's another group of angels who will just figure out what to do with this information. Our goal for now is to communicate as much as we can. Think of those types of neural coding that we studied 2 lectures before. Remember, there was like a rate code and population rate code, and there was a labeled line, there was temporal code, there was phase code. So there are lots of different types of coding. Which ones would be involved here? How would you make this neuron spike? Pause the video, think about that, then continue and compare your answer to the answer in the video. All right. How would you make it spike? So let's think. Different neurons dealing with different frequencies and communicating information about these different frequencies separately, it feels a little bit like a labeled line. So as a whole, the spiral ganglion communicates kind of labeled line style. However, each neuron separately wouldn't necessarily only do spike or no spike. Right? Spiking when it's present, no spiking when it's absent. That's possible, but that's a little bit too simple. What can they do? Okay, we have this oscillation. There are 2 pieces of information in any oscillation like that. Every wave can be described as 3 variables. We have the frequency, and the frequency defines the distance between the peaks. This is technically wavelength, but frequency and wavelength are related. Then there is the amplitude, so how high it is. Amplitude is usually measured from the middle, but it doesn't matter. So amplitude is important. And the other thing is phase. So when exactly peaks are happening. Are they happening like here, as they are in this case, or are they here, or are they here? You can imagine a similar wave that's just shifted a little bit. It's still the same frequency, still the same amplitude, but shifted. Now, if it's just a frequency that happens kind of in the wild, it doesn't matter whether they're shifted or not. But if many frequencies are happening at the same time, or if the sound is fairly short, say it's not a flute playing, but it's more like a chirp by a tasty cricket that you want to eat if you eat crickets, then when exactly these peaks are can matter. So there is the wavelength or frequency, I will call it frequency. There is the amplitude, like amplitude, and then there is the phase. The frequency we don't have to encode because this neuron only deals with one frequency, but amplitude and phase we can. How to encode amplitude? Well, the easiest way is probably go with rate code, right? So let's spike more if the sound is louder, and let's spike less if the sound is quieter. That's good, we can do it. What about phase? Can we encode phase as well? Yeah, we can actually encode it almost like for free, because we can make the neuron spike in synchrony with the signal. Let's agree that every time the neuron spikes, if it spikes at all, if the amplitude is high enough, let it spike around here. Like, what does it mean? This is timeline, t, so time goes, and let's make it always spike in synchrony with the curve. I somehow drew it like it's spiking after the peak. It doesn't matter. It may be like with the peak or before the peak. As long as it spikes in synchrony, one way or another, it will convey information about the phase. Right? Is it hard to make it happen? No, it's actually very simple because look, these guys are moving, right? Every time we have a vibration, the basilar membrane moves up and down, remember this part? And as it moves, if you look at what happens here, my hand, which represents the tectorial membrane, I should probably use my left hand, right? To resemble the picture. So my hand moves relative to the basilar membrane, which means that it moves the hair cells here and back. And every time they go in one direction, they depolarize. And every time they go in the opposite direction, they hyperpolarize. So just naturally, this vibration up and down will cause the tectorial membrane move left and right, which will make the voltage of the hair cell go up and down, and then they release glutamate. Remember, there is a synapse here, so each hair cell, this is like demonic looking hair cell, but whatever. So it has vesicles of glutamate, and it releases glutamate every time it depolarizes. So every time the membrane moves in one direction, the tectorial membrane moves in this direction, the voltage increases, and then as voltage increases, glutamate is released, and as glutamate is released, action potential potential is sent down the axon, which means that spikes are naturally in synchrony with the motions of the membrane. They will naturally occur at the same phase relative to how membrane moves, so relative to vibration. The phase is kind of naturally encoded by the spiking. Will it be able to spike though? I mean, is it fast enough to spike? Because remember, the reason we have the cochlea is because neurons are not fast enough. Each spike takes 1 millisecond for the spike itself, and then a few more for the refractory period when it needs to recharge. So can it do this, what I drew here? Can it spike in synchrony with these oscillations? It depends on the frequency. If the frequency is low enough, that's a low frequency. If it's low enough, then it can spike in synchrony with peaks of this oscillation. What if it's higher? Well, how's about we will just miss some of the peaks. So when we spike, we will still spike, we'll make it spike in synchrony. It will be phase locked, so it's type of phase code. But if on some particular round of oscillations, on one of the peaks of this vibration, it can produce a spike, not a big deal, it's okay, just use the next one. Here's the summary of that. If the oscillation is slow, these up and down, up and down, up and down is happening slowly enough, then let's make the neuron spike in synchrony with the oscillation. The phase at which the spikes happen will always be the same. That totally works to 500 Hz. If we go higher than that, we still spike in phase, but we just miss some of those. So we spike, and then we take our time to recover, but next time we try to spike, we still spike in synchrony with the same phase as we used to spike here. It wasn't a very good sentence, but you got what I mean. It's still phase locked. If the brain is listening, then there is still information here, not just in the number of spikes, which is also there. The more spikes, the louder the signal. But not only the number of spikes matters, but also when spikes happen matters. So we have temporal code as well, which make it a phase code. It's like a population temporal code, but you can see how everything is mixed, right? It's labeled line as far as different frequencies go, but then it's kind of phase code if we start to compare phases of different frequencies, and a little bit of rate code is thrown in because we have more spikes when it's loud. So these types of coding are more like colors on your palette, but you can mix them as you wish when you are trying to design a circuit, like reverse engineer a circuit. So that's what happens up to about 8 kilohertz, which is a pretty high sound. I'm not even sure how to illustrate it. It's like really high whistling. My highest whistling is still about 10 times lower frequency. If we go above that, the vibration is too fast and spiking is no longer phase locked. And basically it's just whether the frequency is present or not. And by the way, these frequencies, whether you can even hear them or not, depends on your age. If you're young, if you're like typical college age student with normal hearing, you totally can hear them. If you're like typical professor's age, you can barely hear them. If you're like retired, you probably cannot. So 8 kilohertz is about at the point where by retirement you probably wouldn't be able to hear it, which makes recognition of speech actually harder, so people get hard of hearing. But we'll talk about it a little bit later. What are the projections in the brain? I'm not even sure why I'm showing this slide, in the sense that it's too much information for us, but let's at least get the spirit of it. The spiral ganglion that's sitting near the cochlea projects to the ventral cochlear nucleus, which is somewhere in the hindbrain, which means that it's not quite in the neck, but pretty close to the neck. It's in the part that's close to where the brain connects to the spinal cord. Then from there, we have two pathways. One pathway basically goes to this startle network. If the sound is sudden, you want to get startled, like you want to get startled. This evolutionary idea that it's nice to get alerted if a sudden sound happened. And that's a dumb network, the network that doesn't analyze the sound, but just gets startled just in case. But then there is a smart network that's going to analyze the sound. The smart network goes into another nucleus, also in the hindbrain, called superior olive. Then it goes to the midbrain, to the part that's called inferior colliculus. And then from there, it goes to the thalamus, to the medial geniculate body, and then to the auditory cortex. Well, so what can we say here? If we are not trying to memorize that, and I think we probably shouldn't at this point, let's just think of some global ideas here. So first of all, there are lots of connections. It's not just one neuron immediately talks to one area in the brain. No, there are lots of connections. That's point number one. Point number 2, there may be different pathways. So in this case, one pathway is smart and tries to analyze the sound. Another pathway just makes you startled if the sound is loud and unexpected. And that's normal in the sensory world, in how your brain analyzes information. One neuron can send accents to several different parts. That's point number 2. Point number 3, in humans, because we have cortex, the most fancy connection goes to the cortex. Then there are connections in the cortex. This is the primary auditory cortex, which is the first part in the cortex that receives this kind of information. But then there will be connections within the cortex. We'll talk about them later. In humans, cortex is probably the highest level. In a bird, it would be some different nucleus. Birds don't have cortices. In mammals, it will be the cortex. In fish, I don't know how fish think, but there will be some part of the fish that analyzes auditory system. Learning point number 3. This connection made a stop in the thalamus. Why is it important? We just happen to have mentioned thalamus before, because for touch, we also had a projection in the hindbrain, then it was a connection to the thalamus, then to the cortex. For gustation, it was the same. It went from the tongue into the hindbrain, different nucleus in the hindbrain, obviously, then to the thalamus, also different nucleus in the thalamus, and then to the cortex. So thalamus is this part through which each sense goes, with an exception of smell, of olfaction, as we discussed 2 lectures before. So thalamus, medial geniculate body, is a part of the thalamus. Let me write that then. Thalamus is a part of the brain that, among other things, stays in the way of all the sensory information that comes to the brain. At least before it reaches the cortex, it needs to show up at the thalamus. So that's a place that can filter information. If you wanted to filter information and make only part of information reach the cortex, for example, if you want to pay attention to something but not to something else, thalamus would be a good place to filter it. Gustation goes through it, touch goes through it, hearing goes through it, will vision go through it? What do you think? We'll talk about it later. Yes, it will. Spoiler alert, it also will go through the thalamus. Okay, that was learning point number 3. Learning point number 4, hearing in particular, makes a stop in the inferior colliculus. This part. Why does it matter? As we'll learn in a few minutes, inferior colliculus answers a question of where the sound is, while cortex in us, in humans, answers the question of what the sound was. So different parts of the brain can analyze different aspects of the signal, and then all of these pieces of information are synthesized together. I guess that would be enough for now. And let's talk about sound localization. So how do we figure out where the signal is, where the sound is, where did the sound come from? Can you figure that out? What do you think? If you hear a sound, like maybe it's here, maybe it's here, is there a difference? With your eyes closed, like assuming, of course, that you have 2 normally functioning ears, because not all people have, that's okay. I mean, that's sad, but normal. Assuming that you have what's called normal hearing, can you figure out where does the sound come from? Yeah, you can. How do you do that? Let's talk about that. Sound localization. Why should we talk about that? Because we kind of know how it happens. Is it the most interesting part of sound analysis? No, it's not. The way we analyze speech, the way we listen to other humans, so the way we figure out from the sound what actually happened, these parts of sound processing are fancier and more interesting. Where the sound comes from is relatively simple, but the trick is we don't actually know, or at least we don't know that well, how we understand what the sound was. Like, how do we recognize voices of our loved ones if we can recognize them? We usually can, right? It's a fancier calculation and we don't quite know how it works. While for sound localization, we kind of figured that out. So let's learn that and at least get some idea of how things like that may happen in the brain. Like, how do you analyze these spikes that are arriving in the brain? Okay, so let's start with basic questions. What aspects of the sound, what properties of a sound can we use in order to figure out where it is coming from? Let's think. So try that. Make some sounds in one direction, make some sounds from a different direction. What could be the difference? What do you think? What properties of a sound can help us to figure out where it's coming from? Pause the video, think, then continue, compare your answer to mine. Got it? How can we figure out where the sound is coming from? We can compare the loudness between the ears. The thing is, our head is heavy and full of fluid or whatever. I mean, it's not exactly water. It's water with some fatty tissue inside that we call brain. But it's pretty heavy, and so it doesn't move when the sound hits it, which means that for some frequencies, it has a sound shadow. It's not the case for low frequencies, actually. Low frequencies just get around the head, but for high frequencies, they cannot get around. They cannot go through because they get absorbed and reflected, which means that if something is happening on one side of your head, one ear will hear it much better than the other ear. When we're in the room, both ears are fairly activated, but if you're in the field, for example, if you've ever stood in the field, a sound produced like that on one side of your head will almost only activate one ear. The other ear will be in the sound shadow. So if you look at how strongly your ears are activated, we can figure out where the sound is coming from. If it's in front of you or behind you, the activation will be about the same. If it's on the side, one ear will be activated stronger. What else? What other information we can use? Another piece of information is inter-aural delay. And this is a little bit fancier, and this thing happens because sound doesn't propagate immediately. It doesn't happen everywhere at the same time. It does propagate. If there was an event that produced a sound wave, the sound wave is traveling with a speed of sound, which is like 300 meters per second, 340. It depends on the air pressure, which means that it's slightly different at different locations, but it's about 300 something meters per second. So it's fast, but not ridiculously fast, actually. Let's think about a spike. Let's look not at the total length of the spike with everything, but the length of the peak part itself. It's about 1 millisecond, right? Approximately. The speed of sound is, let's say, 340 meters per second, approximately. So in a second, it does 340 meters. In 1 millisecond, it will do 0.3 meters or 30 centimeters. 30 centimeters is like size of a ruler. It's that much. In 1 millisecond, which is the length of a spike, sound travels approximately the size of our head. Okay, so it's reasonable. It's reasonable. We can actually hope to notice the difference. It's reasonable in the sense that every sound hits one ear earlier than the other ear by about 1 millisecond, slightly less than that. Oh yeah, it's actually shown here on the picture. It says 0.6 millisecond. This ear is hit a little bit earlier if the sound happened on this side, which means that you can encode it with spikes potentially. These differences are within the realm of what you can encode with carefully placing the spike in time using temporal code. And we've just discussed how temporal code is a big part here because spikes happen in synchrony with oscillations of a sound wave. Good, so the brain has access to two, at the very least, two parts of information. The difference in loudness into ears and the difference in phase into ears. Which ear oscillates first, which ear oscillates second. If the sound is coming from in front of you and from behind you, it will be in phase. If it's coming at an angle or at one direction, one ear will be leading and the other lagging by slightly less than a millisecond. How do we figure out what's high and what's low? And why are our ears so fancy? Why is the shape so fancy? And the fact that I show this picture kind of hints at the answer. Yeah, the only way we humans can figure out whether the sound is coming from below or from above is because it reflects slightly differently from our outer ears, depending on where it comes from. If our outer ears were just conical shape, like those artificial metal constructions that we've seen in the previous lecture, we wouldn't have been able to figure out whether it hits it from above or from below. But because they have this shape, different frequencies are filtered slightly differently. Like it's coming from above, it hits like this part. If it's coming from below, it hits like this part and then reflects. And as a result, they sound slightly differently, but not too much. There is this really nice simulation that you can make to make sounds that, if you listen to them in headphones, sound as if somebody really was moving around your head. And I give a link to one of those in the description. So it's not just somebody speaking close to the microphone. You need a special microphone for that. You need a stereo microphone, but ideally, you want to buy a mannequin and like take the head of the mannequin and fill it with water or gelatin or something like that. But water is probably best. Fill it with water to simulate the consistency of a human head, and then it has to have ears, and then you put microphones in these actual ears. This becomes microphone 1, this becomes microphone 2. And if you record from a thing like that, you will record these same sounds that would have hit real tympanic membranes of a real human. Which means that if you use this giant head-size microphone, you will record sounds as they would be experienced by a human. So if you record like that, create a stereo channel, and then listen to that in headphones, you would hear the sounds as if they were happening around your head. And that's how these funny videos are recorded. And that's how you try to synthesize audio for video games, for example, if you want the sound to be really 3D. And if you listen to those, you can figure out that left and right and around your head, you can kind of figure it out. Up and down are much harder. You can try to force yourself, kind of try to get into the illusion, but up and down is not perfect in humans. And if you use a basic stereo microphone, like the one that just has 2 tubes sticking in different parts, you have probably seen those. They look kind of like that. The fancy, expensive microphones with a stereo microphone. You won't be able to figure out up and down with a recording like that. You really have to have it connected to realistic looking ears. I'm not sure what I'm drawing in order for this thing to work. Okay, primary auditory cortex. If we look at the primary auditory cortex in the human, because different frequencies are detected by different neurons, because the sound is disassembled into this picture where it's represented as a sum of frequencies, the brain just speaks this language. So in the cortex, in the first cortex that receives information from the ear, which is called primary auditory cortex, we have a map. A map meaning that different parts of the cochlea correspond to different parts of the cortex. So neurons that are here that respond to high frequencies would connect eventually to here. As you remember, there are several synapses here actually. It's not one-to-one connection, but eventually the signal will arrive here. Neurons from here, again after several stops, will speak to neurons over here, while neurons that detect the lowest frequencies after same number of stops will speak to neurons over here in the cortex. There is a map. And it's not the first map that we've seen. We've already seen 2 maps, kind of. One was the map from our skin to the somatosensory cortex. The other was barely mentioned, a map of different smells. But this is the map that maps different frequencies or different positions in the cochlea, which is the same as different frequencies, to different sub-areas in the primary auditory cortex. Why maps are useful again? Why maps are a thing? Remember touch, like why your fingers and your palm are projecting to places that are nearby? Because comparing signals from here and from here make the most useful comparison, right? This part of skin and this part of skin are probably transmitting information to the brain that's more mutually interesting, so to say. Like this comparison provides you more information than comparing information from these fingers, like and these elbow. They are in a different part, in 2 different parts of the world, but these 2 guys are in the same part of the world. Think of a little bug running on your skin. That's not a pleasant idea, but imagine that something is moving on your skin. Ew! But it will activate your finger first, and then it will activate the skin of your hand, and then it will activate the skin of your wrist, and then your arm, right? So if you want to reconstruct the movement of this something against your skin, you really want to compare this to that, this to that, this to that, this to that. That's why we have a map that represents our skin in the brain. And the same applies here. If you want to figure out what's happening with the sound, it kind of makes sense to compare neighboring frequencies to each other more than frequencies that are far. I guess it's actually less trivial here. But anyways, that's what's happening. Let's go further. Owls. Let's talk about owls. We just discussed what types of information are available to the brain. Comparison of the loudness in the left and right ear, comparison of the phase of the sound in the left and right ear. But how does the brain work? Why owls? What's special about the owl? Well, that's this champion species principle. Remember, for everything that you may want to study, there is probably an animal that's really good for studying this thing. And owls hunt at night. They have really awesome night vision, that's true, but mostly they hunt by ear. How can you prove that? How can you prove that they hunt by ear if there is a little bit of light? Can you prove that? The experiment is apparently very simple. You just take a mouse and you tie a little piece of rustling paper behind the mouse, and the barn owl attacks the paper, not the mouse. So it hunts using its ears, not using its eyes, and it figures out where the sound is coming from so precisely that it can make the strike with its talons. Because see what's happening in this picture? What's that? It's extending its talons to catch the prey. So it's a pretty precise motion. You have to really catch the prey and not like snap somewhere in the air nearby or run into the ground, right? So somehow using your hearing, using the rustle that the mouse makes, you need to figure out where it is precisely enough to catch it. And barn owls are pretty widespread. They used to be least concern, so they used to be like everywhere. Now, in many states, they are becoming rare because there are fewer barns and fewer farms and fewer places for them to live. They're called barn owls because they tended to live near humans and catch mice that were abundant in the fields and where the grain is stored. But still, you can raise them in the lab, I believe, and some of the most important discoveries about how hearing works were made in owls because their hearing is so good and so easy to study. So how would you study sound localization in an owl in a lab? You cannot really give it mice every time. How can you figure that out? Like, how can you design an experiment so that you could manipulate something about the position of the sound and figure out the responses of an owl? Imagine the lab like that. How would you design this lab? That's a hard question. That's a hard question that I'm asking. But try to pause the video and think and then compare it to the answer. Okay, so what they did is they would put an owl in the dark, and then they would have this giant semicircular contraption with a speaker on it, and the speaker would be moved. So you could move the contraption, you could move the speaker. And these days you wouldn't do that actually. These days you would rather have 1,000 different tiny speakers and control them with a computer, but it was long ago when they did these experiments. And the owl would be sitting there in the complete darkness, and when the sound would sound, it would rotate its head to kind of hear it better. It's like us when we stare at the source of the sound to see it better, right? You can also train the owl to do that by giving it something tasty when it does it, but it also does it naturally. It's a natural behavior. The only thing that you need now is to measure somehow how it turned, but you're in the darkness, so how can you measure it? They put a little electromagnet on the owl, not quite, but something close. So they used something like electromagnets, some coils, to electromagnetically track where the owl is looking. The details here are not that important. What's important is that they could play sounds from different locations and they could see whether the owl looks there or not, like looks, more like hears. We don't even have a word for that in human language, like hearken. Hearken to this sound. Hearken over there. It turns out that the owl can figure out the direction with 1 degree precision. What is 1 degree? It's like the size of your finger if you look at it at an arm's length. If you take your finger and you put it at an arm's length, that's the size of it. So that's the precision. It's very precise. How does it do that? They plugged owls' ears, and it turns out that if you plug an ear, there is a small left to right mistake. It starts to get biased left to right, which is to be expected, right? Because one ear is now plugged, it can't hear that well, so when you compare the left to the right, it feels like this ear gets a little bit weaker signal, so there had to be a bias. But interestingly, the elevation mistake, so looking up, hearken higher, hearken lower is much greater, which totally doesn't make sense. Why would the elevation change? The elevation at which the owl thinks the source of the sound is. Why plugging one ear changes owl's opinion about the elevation of the signal more than the owl's opinion about the azimuth towards the signal? What does it mean? That's a weird question, I won't torture you. If you ever heard something about owls, you may know the answer. If you haven't, it's pretty hard to guess, because owls are weird. many owls have asymmetric ears. And because of that, they use left-right amplitude, the comparison of amplitudes, not to figure out whether the signal comes from the left or from the right, but to figure out whether it comes from above or from below. And for left to right, they use inter-aural delay. That's weird. The way their ears are organized are weird. This is not the barn owl. This is different owl. This is the one with the most asymmetric ears that are so asymmetric that the skull itself has an asymmetric shape. So you can see that the opening for one ear is looking up and the opening for another ear is looking down. And owls don't have outer ears like us. They don't have this pinnae, but they essentially do have outer ears, just they're made of feathers. Around each ear, they have feathers that grow like that, and they can grow in whatever way you can shape the shape of the ear. So owls are weird. They use the comparison of loudness between left and right in order to figure out how high or low things are. And they use the inter-aural delay, the difference in phase, the difference in when the sound hits the right and the left ear, in order to figure out whether the sound is to the left or to the right. It's different from humans. Humans use amplitude for left-right, humans use delay for left-right, and humans rely on really badly functioning slight difference in spectral content for up-down, because humans don't hunt in 3D. We don't fly among the branches to kill a squirrel, to catch a squirrel. We don't do that. We are pretty much living on a plane. We rarely climb up the trees at night. Right? So left-right is important for us, and both systems are dedicated to left-right for us. But for owls, you have to really hear in 3D. So they use inter-aural delay for left-right and comparison of amplitudes for up-down. But so, comparison of amplitudes, that's simple, right? We basically talked about that even as early as our discussions of mouth nerve cells, when the cell would get some inhibition from one side and then some activation from the other side, and that what allows you to compare to stimuli, like who will win, inhibition from the left or excitation from the right, or the other way around. So comparing amplitudes between the ears is easy. But what about the comparison of phases, the inter-aural delay? This is fancy, because the head is not that large, right? In a human, remember, in a human, the delay between the left and the right ear was about 0.6 milliseconds, but the owl is probably smaller, right? If this is the human head, the owl is probably that big. So the difference in when the sound hits the right ear and the left ear is even less than 0.6 milliseconds. It has to be about 0.2 or something. I'm not sure what the number is exactly, but that's the estimation. So it's smaller than the width of the action potential. How can you detect those? How can you detect whether the sound waves activate your left and right ear in synchrony, or whether the one ear lags and the other ear leads, or the other way around? People study that. So what researchers figured out in the 70s is that owls have this special nucleus in the hindbrain, in the cochlear nuclei. They would have a nucleus magnocellularis, like the names are not important, but then there is this nucleus laminaris, where, if you look at it, neurons spike differently depending on where the sound is coming from. So this is the nucleus that detects the direction towards the sound, the left-right direction towards the sound. This is the nucleus that somehow performs the comparison of phases of spiking coming from the right and from the left ear. And they stained cells in these nucleus. Staining a cell means that somehow you perform some magic and one cell becomes black while all other cells remain transparent. And then you can see the shape of the cell. So this is a drawing made from a staining, also in the 70s, that shows how cells project. Here, the cell bodies are sitting over here, and somewhere are the dendrites that come here from the spiral ganglion. And see, it's symmetric. So this is the left side of the brain, and this is the right side of the brain. But they only stain cells in one half. So the cell is sitting here, and then it sends an axon, and then this axon bifurcates. So it splits into two. One half goes ipsilaterally, stays in this half of the brain. The other part of the axon goes contralaterally. It crosses to the other side. And then these axons grow and enter the nucleus laminaris. So they grow like that on one side, and they grow like that on the other side. And so we now have two pieces of evidence. We know the shape of the cell, but also we know that different neurons here are sensitive to different locations of a sound. If the sound is coming from one direction, so this is an owl. If the sound is coming from here, like these neurons over here are spiking. When the sound is coming from here, from in front of you, like these neurons are spiking. When it's coming from the other side, like these neurons are spiking. So where you are in this nucleus corresponds to where the sound is coming from for the owl. How does it work? How can it work? This is a fancy question. Let's do it together. I won't stop the video. Just follow me. Let's do it together. Okay. Different positions in this nucleus correspond to different sources of sound. Sound coming from different directions. Good. The shape is peculiar, right? It goes like that, and then it like connects like that, and it connects in a different way on this side. So let's concentrate on the shape. Let's concentrate on the shape and try to understand what's going on. If you try to schematically draw it, you can notice that on one side, it kind of gets to this point, to this single point over here, and then it goes in different directions from this point. Right? Accents reach this point and then they branch from it to all the possible directions. While on the other side, it's more like branching goes like that. There is one accent and every now and then there is a branch that's coming off it. Real accents are always a little bit twisty and bendy, but generally this is the pattern that we see. Okay, can it be part of the answer? Let's think. Here we look at the cell on one side of the brain, but that's just because we only stained cell on one side of the brain. Obviously, somewhere on the other side of the brain, there is a similar cell with similar accents, and they also branch in one way here, and they also cross and branch in the other way here, right? So let's try to imagine if this cell is on one side, how would the cell on the other side look? Let's hide the brain, look only at the schematic. Now let's reflect it, and this would be the cell on the other side. Interesting. So within each nucleus laminaris, within each of these elongated nuclei, we have accents from the same side and from the different side, entering it from two different directions, and these accents have different shape. It's shaped kind of like a star with several branches radiating from the same point for ipsilateral accents, and it's branched kind of like I'm not sure, like a comb, like a series of offshoots for the accent that comes from the other side. Okay, let's concentrate on that. Let's look at only one of these, and then let's simplify it even further and rotate it and think about what it could mean. Both of these neurons get information eventually from the ear, from the spiral ganglion, right? So this is same side, ipsilateral. This is different side, contralateral. So somewhere there are two ears. Somewhere there is a barn owl and it has two ears. Well, I will draw a human because I don't know how to draw a barn owl. So there are two ears, and one ear is connected to one neuron and the other ear is connected to another neuron. All right? Good. What happens if, say, there is a stimulus and stimulus is right in front of you? In this case, the sound hits both of your ears simultaneously, and so both of these neurons will be activated simultaneously. This neuron and this neuron will be activated simultaneously. What happens then? There are two spikes, a spike in the ipsilateral neuron and the contralateral neuron. Here ipsilateral neuron is on the right. Spikes arrive and then they start to travel. Once they reach any branching point, they obviously branch. So look, if we imagine that these spikes are propagating, this spike is just going forward so far, but the contralateral spike, because there was a branch of the axon, it created spikes at two branches. Okay, they will be at the next point. Like this will travel here, this one will travel here, this one will travel here, then this one will travel here, this one will travel here, this one will travel here. So at some point we'll reach this point, right? Here, for the ipsilateral neuron, the spikes had reached the branching point, and only now they will start to go down every branch, while in the contralateral neuron, this spike reached the extent to which this branch of the axon goes, while this other spike is traveling down and is about to reach the next branching point, and then it will eventually arrive at the furthest branches as well. What will happen at the next stage? Something like that. And now let's look at that. Aha, how interesting. If we look at everything that happened in this lower half, in the lower half, contralateral spike reached it early, but the ipsilateral spike wasn't yet ready. And then ipsilateral spike reached it as well. By that time, contralateral spike was gone. On the furthest branch, the ipsilateral spike reached early, so it's already there, while contralateral spike is just going there. It hadn't yet reached it. But for this branch in the middle, the spikes had met. What does it mean? Because remember what we want to do, we want this nucleus that's shaped kind of like that, cells in this nucleus, cells that are not shown on this picture, cells that are sitting here, like there is a cell here, a cell here, and a cell here, a cell here, a cell here. So we want these cells to spike depending on where the stimulus is coming from. Can this picture make it happen, basically? Can we get sensitivity to the position? Can we have neurons sensitive to the position of the source of the sound from this arrangement of fibers? It looks like yes, we can. Look at that. Just have a cell here that's listening to both of these fibers. Like we'll have a cell here, we'll have a cell here, we'll have a cell here. When spikes arrived at both inputs at the same time, only this cell will spike. If we make this cell, if we like tune it so that it only spikes when it's activated by both fibers simultaneously. You can make a synapse that needs two sources of activation. You can make a cell that needs both synapses to be activated. It's actually slightly different, but we can make a cell that spikes only when both of its inputs are activated. One input is not enough, the other input is not enough. They both need to be active in order for the cell to spike. If you populate this nucleus with these cells, you will get selectivity, because if spikes arrive simultaneously, by the time they propagate through this structure, they will only activate cells here in the middle. But see, like here, why these cells were silent? Because the ipsilateral spike was late, which means that in different situation, when we first activate ipsilateral spike and then we activate contralateral, so if it hits one ear before the other, the ipsilateral spike will come just early enough to meet this spike in time. If we send these two at the same moment, ipsilateral is late. But if we first activate ipsilateral ear, this one and this one will actually meet. And the other way around, if we make this one, the contralateral, a little bit early, start a little bit early, it will be here by the time that the ipsilateral comes. So this part will spike. I'm not sure I'm doing a very good job explaining that. But if you think about this, if you stare at this diagram and remember that these are spikes propagating and meditate for a while, you can figure it out. This is a diagram which basically shows the same thing. If you have this constant delay, so one ear always receives the sound wave a little bit before the other one, and therefore spikes from one ear are always coming a little bit before the other one, you'll have a different part of this nucleus laminaris activated compared to the scenario when both ears are always activated at the same time. And the way to describe that in engineering terms, it's a delay line and a coincidence detector. Why a delay line? Because these fancy accents that are shaped like that on one side and like that on another side, these fancy accents serve as delay lines. Spikes that travel from here will arrive at different parts of these accents at the same moment, but spikes that travel from here will arrive at these dendrites at different moments. So some of them will meet and some of them won't. Does that make sense? Here in this picture it's drawn not like I drew that, but in a slightly exaggerated way. So this is the shape in the barn owl, but you can draw it like that, and then the delays are exaggerated, because then there are delays in both branches, they're just opposite, but it doesn't change the overall picture. And indeed, if you look into the brain of the owl, you will see that it has an auditory map. So again, a map, but not a map that shows different frequencies, but a map that shows different locations. So there is a part in the brain, in the midbrain, that responds to different positions of sound differently. And you can see that when sounds come from one direction, one part of this part of the brain is activated. And when sounds come from a different direction, another part of this part of the brain is activated. And basically then you just wire it to motor systems. Well, this part is not simple, of course, it's easy to say, it's hard to do, but at least we know that the owl has access to this information. It manages to calculate this information from the sound. So you can imagine that some other networks in the brain that are fancier, admittedly, and that we don't understand, somehow translate this information about where the sound is coming from into a motor program about flying properly and extending talons properly and snapping properly that helps the owl to catch a mouse. Good. This concludes this lecture. I hope if you revisit it several times, maybe also read the additional literature that is linked in the description, you can figure that out. If you think about the delay lines, maybe draw it on a piece of paper and just model how the spikes propagate, and imagine that there are cells sitting in different parts of this nucleus that serve as coincidence detectors. You can convince yourself that the system may work, and this arrangement of axons and cells can detect where the sound is coming from. But if it feels like a little bit too hard, don't give up. Yeah, don't give up. Thank you for tuning in, and see you later.
The mystery of hearing.